All right, so thanks for having me. Thanks for being here. I've been, let me move this down here. So like Anissa had mentioned, I had been deployed to the COVID units back in March when the first surge hit us pretty hard. We were actually considered the epicenter of the pandemic at the time. So it was a pretty I was pretty struck by the experience on a number of in a number of realms. One being the virus itself. It's very unpredictable and nothing like we've seen before. But then also, as a psychiatrist, I pay attention to I noticed the fear. There's a very strong sense of fear and uncertainty. It was very striking, and we're still almost a year later. We're still in this, and so through that experience, I was inspired to start to think about different ways in which we can slow the spread and resolve this. I started to think of things on a larger scale, and so it led me to this concept called viral forecast, which is real-time detection and monitoring of known and as yet unknown pandemic-threatening viruses. This picture here depicts how I sort of feel right now. I feel like we're in this dark place and we're trying to crawl out of it. So, I've been working around the clock since March to make this happen. So let's go. Oop. All right, so I'm just gonna go through viral forecasts pretty quickly, almost like a commercial, and then we're gonna go into our current study here at Stony Brook on our detection platform. So viral forecasts, it's the mobile app and web app is an uninterrupted local virus information in graphical format generated by real-time data from personal diagnostics. So in sum, it's like a weather app, but it's actually presenting the movement of virus throughout a landscape locally, giving you risks and risk stratification, and also to follow the spread and slow it and contain it. So we are, within this, we have a platform technology. It's an entirely new approach to detect analytes by integrating electrical and mechanical functionality on the nanoscale. So in comparison to the other point of care diagnostics for SARS-CoV-2 right now, it is the most rapid and accurate. It does not require any sample manipulation, and it's very versatile, so it can be made to detect nearly any virus. We have an incredible group of interdisciplinary team, and the path through the FDA is pretty straightforward. As many of you know, there's the emergency use authorization right now, and this is also affordable and scalable. So what is the problem that we are targeting? So right now, as we all know, we've really scaled up testing massively on a global scale, but still the numbers are rising. So it's counterintuitive. We're doing this in order to slow it down, but it's not working. So why is that? So the reason, I'm pretty certain of this. The reason is the limit of detection. the tests that we're using and the approach that we're doing is just not, it just doesn't work. It's ineffective. So you don't want to be, so these tests, these companies will come out with numbers. They'll present their sensitivity being very high, but the thing is they're only highly sensitive to samples that are highly concentrated with virus. So they're excluding the data from samples with low viral load. So some of the tests actually have a 70% false negative rate. And so we're not getting meaningful clinical and epidemiological data. And that's causing a lot of confusion, and you know, we don't have a hold of it. You may recognize the antigen tests are used pretty widely, sort of like a pregnancy test, the lateral flow immunoassay. The limit of detection ranges between 2 to 20 million virions per milliliter. So that's really advanced disease. The virus has replicated, you know, an absurd amount, and without that, it will not produce a positive result. RT-PCR is another one that's pretty widely used, and that also, you know, there's a number of problems with those, but the limit of detection is 100 to 8,000 virions. So what does this mean? We're missing an entire cohort of people who were recently exposed and don't have too many virions in their system yet. The virions haven't replicated yet. Also pre and asymptomatic carriers. So what am I doing? We need to learn to live with the virus before we can live without it. And to do that, we need to incorporate very accurate testing within our lives, built into our routine very seamlessly, like it's not even there. So what I'm looking to do is develop smartphone integrated virus sensors and mobile app for saliva screening, an option to share results before entering common space. Just gonna move this. So it's a seamless self-screen, and it's built into your routine. And it's to confirm you're not a carrier. And then as a result, you'll gain access to the event, to work, to businesses. And you'll also produce incredible data that's meaningful and can help slow the spread, follow the spread, and generate viral forecasts. So that's the overarching concept. And here at Stony Brook, I've teamed up with Dr. Rafalovich, Dr. Simon, and Dr. Lee, and as well as collaborators from outside of the Stony Brook. So Dr. Rafalovich and Dr. Simon, they've been working on this incredible diagnostic, a very radical approach to detecting analytes, and we are now looking to repurpose it to detect SARS-CoV-2, as well as influenza A viruses, including H1N1 and H3N2, H3N2, and we, you know, it's very promising. It's incredible technology. So, of course, we know about the COVID pandemic, but I just wanted to highlight some of the some of the tests that are being used now, point of care tests. There's the RT-PCR that I mentioned, which requires the nasopharyngeal swab, which is you know, very unpleasant and just not sustainable. And if you, the limit of detection, it actually ranges between, the hundreds to thousands, and the diagnostic time is not practical. It can be anywhere between 30 and 2 hours, 30 minutes to 2 hours. CRISPR is another, it's an interesting approach that has some promise, but it's very expensive, and the limit of detection is inadequate. It also requires the throat swab and takes a while to result. Same with the RT lamp and the field effect transistor, which is pretty more efficient, but is expensive, and it can't scale. It also requires a lot of sample preparation, so it's not user friendly. So there's an unmet need here. What we need is a highly sensitive and specific detection device that can detect low viral load. We need a test that will detect and decipher between very similarly shaped and the composition of viruses. It needs to be able to decipher between them. We need to bypass the mutation issues, of course, which are likely inevitable. And we need real-time results. We can't test someone one moment and then get the result hours later, because it's just too delayed, and it skews the data. And people can spread the virus in between. We need something low cost so it can be scalable. We need it to be sustainable, because this isn't something that the virus isn't going to go away overnight. This is something we need to plan for quite some time. So in order to be sustainable, it needs to be easy to use, and definitely can't have nasopharyngeal swabs. We need easy data sharing, and it needs to be sustainable and have longevity. So, we're dealing with COVID-19 now. There's going to be other viruses in the future, so we need to be prepared. So, to swing over to our study, what we're working on is a completely different approach than what's been known. This is truly a material science engineering approach. This is electric, and it's mechanical. And so what is it? We're using a thin, we're using a silicone wafer, very thin, and we're coating it with gold, thin layer of gold, using a physical vapor deposition. And in doing that, we slice it up so that we can use each slice to act as a sensor. So the surface of this gold-plated chip has a rough surface, and then it has a smooth surface. And in between, in the rough surface, you can see here that it's sort of like a mountain range, and there's space in between the mountains. And so that space in between, the space is, it varies, but there's a very large surface area where pretty much there's tons of different sizes and shapes that we can take advantage of. So the way to really conceptualize this is, this is a good analogy where you have the gold-plated chip. We're going to coat it. I'll talk about this, but we coat it with thiol through a sulfur bond, very strong. And then when you add an analyte of choice, so we choose an analyte, we put it in a mixture, and we expose the gold chip to that analyte, it falls, it just trickles, it just settles down onto the surface of the gold chip, and it creates an imprint just like this. So this processing of the gold chip is called molecular imprinting. So we're doing a 3D molecular imprint of the entire 3D morphological structure of an analyte such as SARS-CoV-2. So like I said, you take the gold chip, you add the analyte, which is in a mixed solution with the thiol. The thiol binds to the gold via sulfur bond. It's irreversible, very strong, and it creates a self-assembly monolayer. Then at the end of that thiol, sticking out are these hydroxyl groups that will bind to analytes that are in the solution. And the binding here is electromechanical. It's electrostatic and hydrophobic interactions. So what does this lead to? You have the analyte binding to the thiol and the gold-plated chip. It creates a voltage change, and that voltage change goes through a potentiometer to amplify the signal, and the readout is the voltage change. And that voltage change will correlate to the presence of an analyte and also the concentration of the analyte, which is amazing. So there are two main things that we look at on the OCP response. Here, once you have this linear start, that means that there's the presence of the analyte. So the analyte is, all the analytes in the sample are falling into their imprints. And then once you hit the plateau, that means all of the imprints are saturated with the analyte. So it either means all of the imprints are saturated, or all the analytes in the sample have landed in the imprints. And so that gives you the concentration. So just to give you an example, Dr. Rafalovich, in a previous study, had validated this approach with CEA, which is a pancreatic cancer marker, amongst another number of different things. So this is an illustration of how this works. Again, you have this gold surface. You add the mixture, and so here's the CEA. The CEA settles on the surface of the chip, and it creates an imprint. Okay, you wash it off. You wash all the CEA off, clean it off. The imprints remain. So then you take that prepared chip, and when you expose that prepared chip to a sample, such as a saliva sample, if it has the analyte, the target analyte that you had prepared it with, you'll get a response. You'll get the analyte will fall into place like a glove. So, like I was explaining before, we have this linear response, which will say yes or no to the presence of the analyte. I'm just going to start moving a little quicker because we're running out of time here. But you have this concentration. Eventually you'll hit the plateau, and you'll be able to quantify the amount of viral load in the sample. This is very sensitive and very specific. So, and there's also no cross-reactivity. So when you when you have a saliva sample, for example, you have CEA in it. You also have a number of biomacromolecules, and when you also add very similar shaped and size molecules to CEA, you will not get this linear response. So these aren't no voltage change, for these molecules. And for this, the limit of detection was 0.2 nanograms per milliliter, which is extremely low. So how sensitive is this? It's actually as sensitive, if not more sensitive than ELISA. And it costs a fraction of the price, of course. It takes really seconds to minutes to get a response, whereas ELISA requires over 10 hours. You barely need any sample for this to work, and it's extremely easy to operate. For the user, it's just one step. You just add the saliva and move on. So,
Matthew, do you want to wrap up?
Yes.
At least 5 minutes per question.
Well, I'll just run over to, so this has been used on a number of different analytes that Dr. Rafelovich has proven. Zika, dengue last year, which are very similarly looking. This was able to, the limit of detection was about 1, which is ridiculous. And it was able to there was no cross-reactivity between the two. Right now, we are looking at influenza A viruses, like I had mentioned, and we have validated it with both H1N1, H3N2. No cross-reactivity between the two using a saliva sample. And the limit of detection as of right now is about 2 to 400. Now we are working with SARS-CoV-2, and we have actually, we've validated this with the spike proteins. So we have spike protein for SARS-CoV-2 versus the MERS SARS spike protein. And that has also, right here, there's no cross-reactivity between extremely similar spike proteins. So, I've done a very extensive competitive analysis. So our next steps, what do we want to do now? So up here on the right, we would like to collaborate with a group that presented last week to use this in sewage, actually, to follow large-scale monitoring. I have actually am now a member of New Lab. It's an incredible hub of innovators and engineers. This group that will likely be collaborating with had back in April, they developed ventilators, very small, compact, all designed it, manufactured it through FDA into the market within 1 month. There's a number of different opportunities that we have here. BARDA, the ENACT, is something that I'm going for. We are in this COVID-19 at Anywhere Diagnostics contest through the HHS. NSF has an opportunity. Also, J&J and BARDA have been in talks with. So BARDA ENACT, this is the, there's four goals that this funding announcement has. And I believe that we can tackle all 4 of them together as a collaborative group between, at the least, the 2 groups that presented last week. The biostatisticians will likely be very helpful. So yeah, that's it.